Well, our first speaker, I'm very honored to introduce a true pioneer and trailblazer in the world of genetics. Dr. Mary Claire King is a professor of medical genetics and genomic sciences here at the University of Washington. And she's a member of the breast cancer research program at the Fred Hutch Cancer Research Center here on East Lake. She's had a very interesting career, and she started out, graduated cum laude from Carleton College in Minnesota. She then went to Berkeley, where she received her doctorate in genetics and postdoctorate fellow in cancer genetics at the University of California, San Francisco. And she was the person who described that there's just 1% difference in the DNA and genome that separates we Homo sapiens from our closest relatives, the chimpanzee. Her primary focus has been in the genetic characterization of different family lines that have breast and ovarian cancer. She was the first to prove that breast cancer is inherited in some families by genomic localization of the gene on chromosome 17. So it was truly a breakthrough. People had known through the years that there were certain types of patients that tend to have breast cancer run in their families, but this is the first time that it was really localized and the genetics proven. And there were a variety of different breakthroughs that occurred that helped her through her career, and she was just describing the incredible collaboration that she had with her colleagues at Berkeley, who were really, truly pioneers in genetics and sequencing. So when you watch CSI and they prove who did what to whom, it was actually Mary Claire King who developed that technology, which is truly amazing. So her other interests: inherited deafness, influences of genetics on mental illness, such as schizophrenia, the human genetic diversity and evolution, and the application of genomics to human rights problems as well as CSI. She is really an internationally recognized authority and pioneer. She's on national advisory committees, which include the Advisory Committee to the Director of the National Institute of Health, the United States President's Cancer Panel, the National Academy of Sciences Committee on Science, and the Commission on the Disappearance of Persons of the Republic of Argentina, and which is an incredible story of immense political intrigue too, that these people who had been disappeared later were proven that their children had been given up for adoption to various government officials when they were whisked away in the dark of night. She's an elected member of the most prestigious scientific societies in the United States and the world, which includes the National Academy of Sciences, the American Academy of Arts and Sciences, the Institute of Medicine, and she's earned 13 honorary doctoral degrees at the most prestigious universities in the world. So without further ado, it's a great pleasure to introduce Dr. Mary Claire King to you this evening. Dr. King?
Thanks very much, Hugh. It's always an enormous pleasure to speak at Mini Med School, in part because there are so many age mates here. And I have to tell you that I have discovered, as of last Saturday, because last Saturday I turned 64, that Paul McCartney's song is a great thing. So, of course, I looked up the words, and I've been humming it ever since. And I also found the interview with him, in which, you know, he's 67 now. So 3 years ago, he gave an interview when he was 64, and he said, "It's not like that at all. It's really," and it's not like that at all, is it? It's really terrific to be in our 60s and to feel like we're in our 40s, and to be able to undertake this kind of work with the young people that have the energy and the technology and the work ethic and the drive to get it done. So what I'm going to do this evening is tell you about some of our more recent activities and adventures in trying to understand why, how, and in whom breast and ovarian cancer susceptibility is transmitted in some families. But we'll begin by going back a bit to the 19th century, when an iconoclastic fellow named Paul Broca, who was a surgeon, what we now call a neurobiologist, an anthropologist, a Huguenot, radical revolutionary, definitely my kind of person, working in Paris in the 1860s, wrote the Traité de Tumeur. And in the Traité, Dr. Broca described, in paragraph form, of course, families on whom he had operated, who were his patients. And I'd simply transferred his paragraphs into pedigree form. And it gives us a stark reminder, in the days before genetics was, of course, known at all, of the sorts of observations, the kind of educated surgical and medical observations that have led this field to be as successful as it was. So Dr. Broca described multiple families. This is one of them. He described this family in which breast cancer appears in 4 generations. Circles are women, squares are men. That's not my decision. A line through a symbol means that the person has died, and a filled symbol for this talk will mean that a person has either breast or ovarian cancer, although on this slide, for a reason I'll explain to you in just a second, I've used filled symbols also to indicate other cancers. So Dr. Broca described, of course, multiple cases of breast cancer in this family. And there are many additional children in the family that I haven't put on the slide. I'm just including the people that he described in detail in the traité. And there's a couple of interesting things. First, we notice, of course, that there's a vast number of breast cancers in the family. Second, that they occur at young ages, especially for that time and place. And next, that we see instances in which he says that he finds cancer in the liver, here and here, here in the abdomen. And he writes in the traité, and this is all from autopsy, of course, he says, I'm seeing cancer in the liver of these patients, but he says, I think these are not cancers of the liver. I wonder, he says, if these may have invaded from the ovary. And for this, Broca should get a gold star, another gold star, because now it's clear that the same genes that are responsible for greatly increased susceptibility to breast cancer in some families are also responsible for increased susceptibility to ovarian cancer in the same women. And we'll hear much more about that in a moment. My friend Dominique Stoppa-Lionnet at the Curie has tried to follow forward in time Broca's families, thus far without success, but she's still working on it. I like to imagine what it would have been like if Broca, working in Paris, had met Darwin, working in Britain, and Mendel, working in Plano, all at the same time. And had these fellows ever gotten together, had they had email to communicate, it wouldn't have taken the next 130 years to take the next step in this, which was the identification of 2 genes that are responsible for that inherited predisposition to breast and ovarian cancer. So BRCA1 and BRCA2 were 2 genes that were identified by an approach called positional cloning, which is an approach that we used before we had the whole genome sequence laid out before us, so that now all we have to do is flip on the public browser, which anyone can use, and anyone in this room can use this, figure out what genes are interesting for a particular trait, and go for it. But that was not true in the 90s. And so with enormously painstaking piecing together hand-read bits of sequence, bit by bit, 300 base pairs at a time, BRCA1 and then BRCA2 were identified sequence and mutations found in them. These genes are young. They are conserved throughout mammals, but one finds only the most wispy signs of them in other taxa, which is very unlike most genes that are critical to normal repair of DNA. Most of those genes are conserved all the way to yeast. These genes are not. What are the families like in which we find mutations in these genes? Because it was working with these families that led to the identification of BRCA1 and BRCA2, and the paradigm for that identification is that one finds in families breast cancer in many individuals. One traces genomic regions that are shared amongst the women with breast cancer, but not shared by women who lived to an elderly age without developing breast cancer, with exceptions as we're about to see. And one then seeks to define that chromosomal region in the narrowest possible way. And that took a lot of effort and a lot of competition among many groups, but ultimately when BRCA1 was identified, it was immediately clear, I mean within 18 hours, that this gene would explain many families with inherited predisposition to breast cancer, as illustrated by family 1. So Broca's family was family 0, and this is family 1. Family 1 is, as you see, a family in which breast cancer appears in multiple generations. Breast cancer appears again at extremely young ages in this family. It is sometimes inherited through fathers, half the time. And there are a number of interesting features about the phenotype, about the illness that I want to share with you, and then we'll talk about the genotype, that is the mutation in the gene. So the phenotype, young age at onset, sometimes ovarian cancer also. In the men in this family, but not in all the families that I've worked with, there's an increased incidence of pancreatic cancer. And statistically, there is an increased incidence of pancreatic cancer amongst BRCA1 and BRCA2 mutation carriers. But also, there are women in this family who develop breast cancer who don't have a mutation. This person, and someone else that you can probably see more easily than I can, this person, who have, as you see, slightly older ages at onset than the women who did develop breast cancer. There are also in this family, or is also in this family, one person who lived to an elderly age. She ultimately died in her late 80s, who, despite carrying the mutation of her family and despite her daughter dying of breast cancer, she herself did not ever develop it. The reason is, at the molecular level, that one mutation in and of itself does not cause cancer. You'll never meet a more healthy, productive group of people than women and men with BRCA1 and BRCA2 mutations. Such individuals develop breast cancer in the case of women and occasionally men, or ovarian cancer in the case of women, or very rarely pancreatic cancer in the case of women and men, when, but only when, a second mutation occurs that is specific to the target organ, that is specific to the epithelial cells of the breast or the ovary or the pancreas, so that two hits are necessary for the cancer to develop, one inherited and one somatic, that is one at the level of the soma or the affected organ. So each of these individuals who's developed breast cancer has had that second hit in the same gene, in the BRCA1 gene. This woman was fortunate, and she never developed that second hit. What's the inherited hit? In this family, it's a deletion of two base pairs just in the middle of the BRCA1 gene, where this arrow is, that leads almost immediately to a stop. And two base pair deletions lead to stops because, of course, DNA is written in triplets. DNA encodes amino acids in triplets. So if you think of a sentence like, the fat cat saw the dog run out, that's a sentence all of triplets. And if you take two letters out of the middle of that, you would have the fat cat saw, suppose T and H were gone, you'd have the fat cat saw E, D, E, G, R, U, and so on. You'd have nonsense after it. It's exactly what happens in this kind of situation. This family has a different mutation, and I'm showing you this family to make 2 points. First, that there's an enormous range of mutations in these genes, and we'll see a display of that explicitly in just a moment. But second, to point out that this mutation, which is at the very end of the BRCA1 protein, is enough to wreak havoc. One does not mess with this protein. And that realization clinically in this family led to the ability to determine what the partners, the protein partners of BRCA1 were at this far end of the gene. So the gene starts to be made into protein here, begins with this ring domain about which more in a second, passes through the middle section. There are various mutations that lead to stops in various families. And then this mutation, which leads to a stop only 11 amino acids before the end of this very long 1864 amino acid sequence, but that's enough because that precludes the proper binding of all of these proteins to BRCA1 into a complex that enables cells to transcribe their genes normally, to make normal RNA and then protein off of the DNA sequence. I want to show you this family to make 2 points. One point is that not all mutations in BRCA1 and BRCA2 are little deletions or insertions of base pairs, although the vast majority are. This is a base pair substitution, just one base pair for another, which changes one amino acid, and this led to a big challenge. Why does this base pair substitution matter when others don't? The other point I want to make is a clinical one. In this family was brought to our attention by this woman who came to us in the 1980s because this cousin had just died of her breast cancer. This sister had died some years earlier. And this woman At that time, we had no genetics on the family. This woman wondered if she should have a prophylactic mastectomy and a prophylactic oophorectomy simply based on her family history. And at that time, we were trying to find BRCA1, but we hadn't found it yet. So we made our best assessment on the basis of the chromosomal localization that I told you. We told her that. We told everyone in the family everything about it in great detail. And she decided, partly on the basis of the family history itself, just the empiric appearance of breast and ovarian cancer in the family, and partly based on the logic that if we were right, which of course was a huge if we were right that there was such a gene, and if we were right about where it was, if it existed, then she would be susceptible. She decided to have a prophylactic mastectomy at the age of 32, 8 years later a prophylactic oophorectomy at the age of 40. Then when she was 44, BRCA1 was cloned. We immediately sequenced BRCA1 in this family, and indeed within 18 hours we were able to tell her, yes, she did have a mutation. She's doing very well now, of course, in her 60s. And we were also able to tell her 3 sisters that they did not carry the mutation of the family. However, over here, this woman was 18 when I first met her. And we learned in the same way that she was likely to be susceptible. And she was so young that everyone, including me, we all said, you need to be followed. And she was followed extremely well by a very good gynecologist, a very good internist. And when she was 38, an interval lesion was detected. She was having mammograms, and absolutely state-of-the-art mammograms for the time, every 6 months. And nonetheless, in the interval between 2 of them, a very small lesion, less than a centimeter, was identified. It was already stage 3. It had already metastasized, and she died within 14 months of that cancer. We were, you can only imagine, we were all completely devastated. It said to us, one has to be extremely alert to how aggressive these cancers are, even in very young women, indeed especially in very young women. This mutation in this family led to the illumination of a beautiful part of the BRCA1 protein. So let me go back. This part of the BRCA1 protein is called the ring domain, which it was named by a bunch of Brits, in a totally different protein. It's a cysteine histidine motif that binds zinc. And it had been seen when BRCA1 was first identified, we all ran to the sequence and tried to find out from the sequence what the function might be. This relationship between structure and function is, of course, one of the paradigms of the way we work in genetics. And there was nothing in BRCA1 that had been seen before. You know, I told you these are young genes, except for this ring domain. So, of course, I went looking for ring domains everywhere. And at that time, in the mid-90s, there were, maybe half a dozen ring domains described. And I thought, this is going to tell me what this protein does. And it turns out ring stood for really interesting new gene. So how much did that tell me? We now know what the ring domain does, thanks to Rachel Clevitt here in structural biology. Rachel and Peter Burdovich and her group, using information from the family that I just showed you, determined that that red part of BRCA1 that was at the beginning of the protein, which is this beautiful double coiled region here, and has in its middle these 2 zinc binding bits here and here, partners with another very similar protein called BARD1, which has an equally beautiful pair of coils here, and also 2 zinc finger binding regions here. And mutations that abrogate these binding domains. The one I just showed you is this one with the white star here, another one here, and some, but not all, mutations along these spines that abrogate the pairing of the 2 proteins lead to a loss of the critical enzyme activity of this complex. And the critical enzyme activity of this complex is to bind to a piece of the estrogen receptor and prepare it for normal processed degradation. And if that isn't done properly, apparently what happens, the rest of this is still unknown, I'm making this up, but apparently what happens is that the estrogen receptor and its signaling stays on too long. So this particular mutation basically keeps a cell healthy that shouldn't be in the breast epithelium, the ovarian epithelium, the estrogen-fed tissues. All, again, genetics informing, as Rachel would say, real biology. I show you this family because this is a family with a mutation in BRCA2. And I want to make the point that men with BRCA2 mutations do sometimes develop breast cancer. This is the most extreme example of that. I've chosen it because for its dramatic effect. There are, how many? 1, 2, 3, 4, 5 gentlemen in this family who develop breast cancer. There is absolutely nothing wrong with them in any way except for their breast cancer predisposition. They are all fertile. I haven't put their children on here, but they are all fertile. There's no mosaic Klinefelters, no chromosomal abnormalities. All of their sex hormone binding globulins are just fine. I mean, everything about their biochemistry is fine, except that they have a high risk of breast cancer. That's not true across the board in BRCA2 families. And one question still in our field is, why in some families do men who have BRCA2 mutations develop breast cancer, and in other families they do not? It doesn't appear to be the specific mutation itself. This happens to be a mutation of Dutch origin. If you tell me a BRCA1 or a BRCA2 base pair, I can tell you where in the world that mutation is from. I mean, to the town, and often to the, at least to the century for sure, and sometimes to the decade. These are young mutations, most of them, with a couple of exceptions, most of them are, well, 50 to 400 years old, and they all are localized in time and place. There's 3 in 3 different glacial valleys of southern Sweden. There's 1 each along several villages up the coast of Norway. This one happens to be from Utrecht, although the family's from Berkeley. I promised to show you a dramatic example of how many mutations there are in BRCA1 and BRCA2. There are lots. So here's the BRCA1 cartoon that you have seen before along here, from the beginning of the gene to the end of the gene. Here's the BRCA2 protein from the beginning to the end of the gene. And every one of these vertical lines represents a different mutation in a different family. So there's more than 1,000 in BRCA1, more than 1,000 in BRCA2. This has led to, or this led to, both a technical challenge and an economic reality of identifying mutations in these genes. It's fine if you live in Utrecht. You have a pretty good guess what the mutation is going to be in a family that presents at high risk of breast or ovarian cancer in that place. If you're in Lund, Sweden, you have a pretty good idea. If you're in Molde, you have a pretty good idea. If you are Ashkenazi Jewish, you have a pretty good idea. But if you are an American mutt, like I'm an American mutt, you do not have a good idea, and it's necessary to sequence all the way through BRCA1 and all the way through BRCA2 to find out what your mutation is. And indeed, many of the mutations are private. This was not anticipated by the company that ultimately cloned BRCA2, by Myriad Genetics. They thought that they would have a cystic fibrosis-like situation in which, at that time, although it turned out not to be true either, it was perceived that virtually everyone with cystic fibrosis would have one mutation. Wasn't true, but it was the early thinking about that gene. And that they would have a tremendous profit because they could type everyone, genotype everyone efficiently, quickly, and cheaply. No, it's expensive, it's hard to do, it's very hard to get right, and it's led to a real difficulty because the technology has improved to get it right, but the tension between maintaining a monopoly that's based on the patent of a process versus being able to use new technology that would make that process, make that genotyping better and cheaper, has been a real issue. But in any case, it's all based on the absolutely real, proven biological observation that there are lots of mutations in each of these genes. And if a woman were to come in today, visit Dr. Elmore, be referred to me for determination of her BRCA1 and 2 status, we sequence both genes, the chances are probably a third it would be a mutation we'd never seen before. So there's lots of private ones, even in people from the glacial valleys of southern Sweden. How much of breast cancer in America is attributable to inherited mutations in BRCA1 and BRCA2? Some nice work was done by Elaine Ostrander and Kathy Malone when Elaine was still at the Hutch. This was a few years ago. She's now at the NIH. What these gals did was do that painstaking sequencing through BRCA1 and BRCA2 for about, I think it was about 1,400 breast cancer patients who were selected not because they had a family history or were particularly young, but simply because they were breast cancer patients anywhere in the greater Pacific Northwest area. And they were all ages, and either black or white, and their conclusions were that 5% of all of the patients that were seen in these in this collaborative in this collaborative group, which were many different hospitals, you can appreciate. 5% of patients had BRCA1 or BRCA2 mutations detectable by the strategies that they used, and that's probably missing 15 to 20% of mutations. So it's probably actually 6%. Among patients diagnosed younger, as you would expect, there's a slightly higher, or significantly higher fraction of patients who carry such mutations, and among patients diagnosed older, there's a lower fraction. Among women who have a family history of breast cancer at a young age in a mother or a sister, the proportion is still higher, and among women who have a relative with ovarian cancer, the proportion is still higher. People say, correctly, gosh, that's not a lot of the breast cancer in America. It's true, but it's 10,000 new cases a year of breast cancer in America, disproportionately in younger women. It's also worth pointing out that there has been an incorrect perception abroad in the land for a long while, and that is that BRCA1 and BRCA2 inherited breast cancer are somehow predominantly a Jewish disease. That's not correct. What is correct is that, just like in southern Sweden and western Netherlands and so on, there are specific mutations in the Jewish population. And there has been very complete, successful genotyping in New York, in Chicago, in Los Angeles, here, San Francisco, in the Jewish community, and of course in Israel, nationwide, for mutations that are specific to the Ashkenazim in these genes. So that success has led to the incorrect conclusion that somehow it is a Jewish disease. It's not. The proportion of breast cancer in the Jewish population attributable to BRCA1 and BRCA2 is a little higher than these figures, but not astonishingly so. This is a universal phenomenon. The mutations occur in all populations. These figures hold for both black women and white women. The risks to modern American women of breast cancer associated with mutations in these genes are extremely high. The risks are 20% by age 40, 55% by age 60, and 80% by age 80. There was controversy because, there's controversy about everything in this field, but there's controversy about this result because while it was consistent with the perception of many clinicians working now with modern American women, it seemed not consistent with observations from the literature in which women who turned out to have BRCA1 or BRCA2 mutations upon sequencing didn't necessarily always have relatives who developed breast cancer. So the thinking was, gosh, some women are in a situation like this. Here is a woman who has breast cancer. She has a BRCA1 mutation. Here's a woman who has breast cancer. She has a BRCA2 mutation. 50% of our patients are like this in that they have a mutation, they have an invasive breast cancer, but neither their mother nor their sisters have breast cancer at all. So the thinking was, golly, maybe the risks aren't that high. Why do we see non-familial breast cancer among women with mutations in these genes? It's because some families are at high risk and others are not? No. It's because some families are at high incidence and others are not. Don't forget about Mendel. He was out there working with smooth and wrinkled peas, and all that applies exactly to this as follows. These genes are Mendelian genes, just like smooth and wrinkled peas. BRCA1 is on chromosome 17, BRCA2 is on chromosome 13. Everyone inherits a copy of chromosome 17 from their mother and a copy of chromosome 17 from their father, and the same thing with chromosome 13. These are what we call autosomes. They are inherited absolutely symmetrically from the two parents. Therefore, working backwards, if a woman has inherited a mutation, a variant, from one of her parents, what proportion of the time is it going to be from her father? Half. 50-50 chance that she got that variant from her father, 50-50 chance she got the variant from her mother. If she inherited that mutation from her mother, there's a very good chance that her mother developed breast cancer. We saw that one exception of the 181-year-old woman who had not, but she's really an exceptional case. But if the woman inherited the mutation from her father, with the exception of those remarkable families of which I showed you one example, with BRCA2 mutations, by and large, the men are just fine. So that's what, in each of these cases, that's what we see, is that a sufficient explanation for why we do not see breast cancer in the families? No, but I'm about to give you the rest of the story. So paternal inheritance, here and here. Small families. If there were 12 sisters in these families, there's a pretty good chance somebody else would have inherited the mutation from the father. But with small families, which of course most modern American families are, there's a perfectly reasonable chance that by Mendelian luck, none of the sisters have inherited the mutation and therefore are at the background, general, non-trivial, population risk of breast cancer, which is what we see here. So each of these women had a 50-50 chance of inheriting from her father. There's a lot of 50% in this slide. The mutation. She did not. She did not. Both were lucky. And they are at background risk of breast cancer. So we worked this out systematically, looking only at families like this and then extending them out and looking at second cousins and third cousins and so on. You can imagine all the chasing down we did. And when one does that, one finds that in these families in which the original nucleus of the big family was like the ones I just showed you, that is, had no breast cancer or ovarian cancer in the immediate relatives, if you go out, the women who actually inherit mutations, who are the cousins and aunts and so on in these families, have the same horribly high risk of breast cancer that I showed you for families generally. So it's not the family, it's the mutation. It's also when one lives. The other observation that colleagues made is that this extraordinarily high risk of breast cancer didn't seem to be uniform if one went back in time, and indeed didn't seem to be uniform around the world. There's a very interesting mutation from the Ivory Coast. In the Ivory Coast, the risk of breast cancer associated with carrying that mutation, which is an absolutely devastating wipe out the function of BRCA1 mutation, is not as high as it is among the descendants of the family from the Ivory Coast who live in America. Why is that? It's for non-genetic reasons. So looking only at American women from one study that we did, here is the story that we saw. The risk of developing breast cancer among BRCA1 or BRCA2 carriers who were born recently, after 1940, is shown here and is extremely high. The risk of developing breast cancer among women born before 1940 is shown here. So the curve has been pushed to the left. And all these women are in the same families. They have the same mutations. It's not the mutation. It's not the other genes in the family. What it is, we think, is just changes in our lives as women that have made all of us more susceptible to breast cancer, whether or not we carry these mutations, and those same non-genetic risk factors, all of which lead to much higher levels of estrogen for longer periods of our lives, which make us extremely productive, both cognitively and reproductively, also increase breast cancer risk. That was already very well known for women generally and seems to apply to women with inherited mutations in BRCA1 and 2 as well. The story is different for ovarian cancer risk. In this case, the risk of ovarian cancer associated with a mutation in BRCA1 is much higher than the risk of ovarian cancer associated with a mutation in BRCA2, and that has not changed over time. The next several slides illustrate the importance of horrible and invasive but necessary surgeries among women who carry these mutations. This study was a study of women who decided to have or not have a prophylactic removal of the ovaries and fallopian tubes when they learned that they had a BRCA1 or BRCA2 mutation. So women were being seen in their late 30s. This was from a collaborative group all over North America. And some women who learned that they had a BRCA1 or BRCA2 mutation decided to undergo this salpingo-oophorectomy, removal of the ovaries and fallopian tubes. No one on this slide had any symptoms of any ovarian cancer at the time. And some women decided to have careful surveillance but not have the surgery. And the slide indicates, the graph indicates the difference in the cumulative proportion of women who survived with, or who were continuing to survive without either breast or gynecologic cancer over the next 7 years. And as you can see, there is a very dramatic difference. An analogous result was demonstrated for women who decided to either have or not have prophylactic mastectomy before BRCA1 and 2 were identified, but in consequence of the empiric amount of breast cancer in their family. So this study was done, again, across multiple hospitals, and this goes back years before BRCA1 and 2 were identified. But when confronted with a familial risk of breast cancer, some women decided to have prophylactic mastectomy, obviously an extremely dramatic step to take, and others decided not to. After the fact, years later, tissues from those surgeries were genotyped, and it was determined whether the women had or didn't have mutations in BRCA1 or BRCA2. So everybody on this slide has a mutation. So what the investigators then did was go back to their list, and they said, okay, we now know which subset of women out of all the women who had these surgeries carried mutations in these genes, and let's see what happened to them. So among the women who had a mutation in BRCA1 or BRCA2 and chose to have mastectomy, there were a total over 40 or 33 years, I believe, of 2 breast cancers. In each case, they were up under the lymph node and hadn't been removed at the time of no, one was under the lymph node, one was in the nipple, because the nipple hadn't been removed in that surgery. And among women who decided against the surgery, you see the result here. Another result showing the same thing. Here we have in surgery and gynecologic oncology here, a very active program of talking with women, women deciding what they want to do and when they want to do it, and the consequences have been very successful to women who find that they have mutations in these genes. These are the recommendations, which you do have, that were published first several years ago and then most recently in 2005 for women who carry mutations in BRCA1 or BRCA2. An annual mammogram and MRI beginning at a very young age. These are for the very highest risk women. Discussing the option of risk-reducing mastectomy and recommending risk-reducing removal of the ovaries and fallopian tubes between ages 35 and 40 or upon completion of childbearing, even knowing that that will entail, for most women, short-term hormone replacement therapy, and recognizing that that will provide some increased risk of breast cancer. But the amount of estrogen that's removed in removing the ovaries is much greater than the amount one needs to replace. So it's a fair trade-off. And I'll end here saying thank you to my lab and to the many different private and public agencies that have supported our work. Thanks very much.
Thank you very much. That was excellent. We have a few minutes for questions. If our speakers want to come up, if anyone, please use the mics in the aisle.
Ovarian cancer is another one that seems to be hard to detect until it's pretty late in the process. CA-125 blood test, any comments on how helpful that is, or the ultrasound?
Yeah, so I'll just speak out of turn here. There are tests that we do for gynecologic cancer, uterine and ovarian cancer. They are not ideal at all. There are high-risk clinics here at the University of Washington and other locations too. The standard of care for those is to do a vaginal ultrasound. CA-125 can be positive. It tends to be positive late in the game, so it's probably not the test you want to rely on if you have a strong family history. And that's probably why the prophylactic surgery that Dr. King was describing is so effective, because surveillance for ovarian cancer, even under the best of circumstances, is not ideal.
Dr. King, any comments?
I'd point out only that ovarian cancer, when caught early, is as survivable as breast cancer when caught early, which is to say very, very survivable. But whereas breast cancer is very frequently caught early, ovarian cancer, with the exception of women who have BRCA1 or 2 mutations who have the prophylactic surgery, with the exception of those women, is almost never caught early. So it's a matter of how far the cancer has progressed when it's diagnosed.
Another question?
Dr. King, you mentioned that you could even go to the day with some of the points where this abnormality started. And I was just curious, when you talked about Mendel and Darwin getting together and having dinner together. Is there some historical reason that they think that we've had this happen 400 or 500 years ago? As to what precipitated?
When mutations occur at random.
Right.
And there were population expansions with urbanization, beginning probably 700 years ago, and then with industrialization much more rapidly. So we see more different rare mutations among people generally now because there are simply more people.
Are we learning how we can mitigate them? Are we learning anything from that as far as mitigating those situations?
Well, mutation rates are random, and the vast majority of mutations have no effect on health at all. They're completely neutral. But what we're now learning in consequence of having very good new technologies for being able to do sequencing of entire genomes is to be able to distinguish the vast majority of mutations which are neutral from the occasional ones which have the kinds of effects that we've all been talking about.
Question on this side?
I suppose it could be considered kind of related to the previous question. There's been a fair amount of attention on epigenetics in the last few years, and I know it's kind of speculative to talk about it in specific, but I'm interested in hearing your thoughts about the extent to which we'll be able to sort of kind of draw a line between epigenetic control and how it relates to the genes and mutating, and then from there to how it may precipitate disease. So any thoughts about epigenetics and how we deal with it?
Okay, the question is, what about epigenetics and how does that affect us? So epigenetics is like you could have a normal gene, but there are things that can bind onto a gene, little chemical compounds like methyl groups can bind onto a chromosome and then make that gene behave differently than it would otherwise behave. So sometimes we have problems with our genes doing their job, not because their proofreading is wrong, their DNA is mutated, but because something has occurred, it's binding onto the gene and changes that function of that gene. And so these changes are thought to occur in part with aging, like methylation occurs with aging, but it can also occur under a variety of settings like chronic inflammation and exposure to a variety of environmental bad things. They can cause cancer. There are certain colon cancers that are associated with methylation of certain genes. They tend not to be inherited. These tend to be sporadic cancers. So you can get we talked today about Lynch syndrome, that colon cancer you get on your right side. It turns out that sometimes people get that and it's because the gene is not mutated but it got methylated. And those people don't pass that down to their children. It's simply that they were unlucky enough to have this methylation occur on the gene that normally, if it was mutated, could cause Lynch syndrome. But then they don't pass that down through their family if it's methylated.
Great.
Thank you.
That's near and dear to my heart. Dr. Lynch, of course, being at Omaha, and he studied people in Douglas County, Nebraska, that's my home state, which is also the largest producer of finished beef.
And ice cream.
Yeah, and ice cream, subsequently ice cream, yeah. So the association with certain dietary things as well. Well, I'd like to thank our speakers tonight. This was an excellent presentation. Give them a big hand.